I hope you have seen my videos on carbon and polymers. Carbon and polymer chemistry is the build-up to biochemistry and its sister fields of molecular biology and genetics. All living things that we know of are built and function by carbon chemistry and especially polymer chemistry. DNA, the information storage code for life that is carried in every cell, is a polymer, a giant carbon chain with attached nitrogen and phosphorus groups. Biochemistry is the area of intersection of chemistry with biology. The microscale biological sciences include biochemistry, molecular biology, and genetics. All these fields assume the existence of life and are studies of life processes and products. These fields of sciences are related, but can be distinguished by their definitions as found in the Wikipedia article on molecular biology. Biochemistry is the study of chemical substances and chemical processes that occur in living organisms. These chemical substances include proteins, lipids or fats, carbohydrates, and nucleic acids. Molecular biology is the study of molecular processes behind biological phenomena. The focus of molecular biology is on synthesis, modification, mechanisms, and molecular interactions that occur in living organisms or are the result of living organisms. Genetics is the study of how gene differences affect organisms. Genetics attempt to predict how genes, how genetic interactions, and how genetic mutations will affect gene expression such as the structure, development, or behavior of living organisms. Another way to link these scientific fields is to say that biochemistry is the study of how proteins affect function. Molecular biology is the study of how genes affect proteins. Genetics is the study of how genes affect function. If the intersections between the sciences are not well defined, it is because these sciences overlap. Biochemistry focuses on the composition and function of the four major organic chemicals that are found in all living organisms: lipids, carbohydrates, nucleic acids, and proteins. Lipids are most associated with energy storage and as building blocks of cell membranes. Carbohydrates are most associated with energy. Nucleic acids are most associated with information storage and information transmission. Proteins are most associated with cell structure and cell metabolism. I will look at the chemical composition of these substances first, and then at their functions. According to biologydictionary.net, a lipid is a fatty, oily, or wax-like compound that is insoluble in water or hydrophobic. Some lipids are amphiphilic, meaning one end is polar or hydrophilic, and the opposite end is nonpolar or hydrophobic. Oils are liquid at room temperature, fats are semi-solid at room temperature, and waxes are solid at room temperature. There are three major classes of lipids: glycerides, sterols, and phospholipids. Lipids are composed of glycerol and fatty acids. Glycerol, or glycerin C3H8O3, is 3 carbon alkanes with 3 hydroxyl groups OH. Glycerol is just propane with 3 hydroxyl groups OH substituted in for 3 hydrogens. A fatty acid is a compound composed of a carboxylic acid COOH substituted into an aliphatic chain. An aliphatic chain is a hydrocarbon chain. The simplest aliphatic compound is methane CH4. Fatty acids generally have an even number of carbon atoms between 4 and 28. When the carboxylic acid is joined to the aliphatic chain, the compound becomes a fatty acid. The lipids that we most commonly call fats are triglycerides. Triglycerides are compounds composed of 3 glycerol molecules bonded to 3 parallel chains of fatty acid. If the carbon chains of the fatty acid are all single bonded, the fat is called a saturated fat. If the fatty acid has at least one double or triple bond between carbon atoms, the fat is called an unsaturated fat. If the fatty acid's carbon atoms have multiple double or triple bonds, the fat is called a polyunsaturated fat. Unsaturated and polyunsaturated fats have a lower melting point than saturated fats. An unsaturated fat or polyunsaturated fat that is liquid at room temperature is an oil. Oils can be hydrogenated or have hydrogen atoms added or substituted into the fatty acid structure. These hydrogenated oils raise the melting point of the oil and so make it solid at room temperature. Hydrogenated oils can be used as butter substitutes or margarine. Most fatty acids are cis fats, meaning their functional groups are on the same side of every carbon in the chain. If any of the functional groups are on the opposite sides of the carbon spine of the chain, the fat is said to be a trans fat. Hydrogenation of unsaturated fatty acids has been found to create trans fats. An example of a cis molecule and a trans molecule are shown in the figure. In cis-butene, the methyl groups CH3 are on the same side, above the double bond. In trans-butene, the methyl groups CH3 are on opposite sides, above and below the double bond. In the cis-fat, the hydrogens are on the same side on both sides of the carbon-carbon double bond. In the trans-fat, the hydrogens are on opposite sides of the carbon-carbon double bond as shown in the figure. Sterols are also lipids. The most famous sterol, cholesterol, is necessary to messaging in brain function and the structure of cell membranes. A human body can manufacture cholesterol from other lipids. If too much cholesterol is ingested with food, it interrupts the body's natural formation of cholesterol. Phospholipids have a hydrophilic phosphate head attached to a glycerol with two hydrophobic fatty acid tails. The structure of phospholipids makes them amphiphilic, hydrophilic at the phosphate head and hydrophobic at the fatty acid tail. The amphiphilic nature of phospholipids make them good construction materials for making cell membranes. Carbohydrates are the molecules that make up sugars, starches, and cellulose. Carbohydrates are compositions of carbon, hydrogen, and oxygen, and are generally, but not exclusively, in a 1:2:1 ratio. Carbohydrates are generally called saccharides, from the Greek word for sugar. Saccharides can be classified into 4 groups: monosaccharides, disaccharides, oligosaccharides, and polysaccharides. Monosaccharides are simple sugars that include glucose, fructose, galactose, and glucosamine. Disaccharides are compounds of 2 simple sugars. 2 simple sugars can be joined by a glycosidic bond. These double sugar compounds include sucrose, lactose, maltose, trehalose, cellobiose, and chitobiose. Oligosaccharides are compositions of 3 to 10 monosaccharides. Polysaccharides are long-chain sugars normally associated with cellulose and starch. Starches are found in the human edible parts of plants, including seeds of grain cereals, and starchy root vegetables such as potatoes and yams, as well as other seed foods such as beans, peas, and lentils. Cellulose is found in the fibrous parts of plants such as stems, leaves, and tree trunks. Cellulose is indigestible to humans, but some animals such as cattle and insects such as termites have bacteria in their guts that can reduce cellulose into simpler sugars that are digestible by their hosts. There are 2 types of nucleic acids, DNA and RNA. RNA can come in 2 types, messenger RNA or mRNA, and transfer RNA or tRNA. DNA stands for deoxyribonucleic acid. RNA is ribonucleic acid. DNA has the form of a double helix. RNA is a single helical strand. Nucleic acids are composed of nucleotides. Nucleotides are composed of a 5-carbon sugar, a nucleobase, and a phosphate group. The phosphates and sugars make up the spiral spline of the nucleic acid molecule. The nucleobases make the rungs or treads of the spiral ladder of the nucleic acid molecule. In RNA, the sugar is ribose C5H10O5. In DNA, the sugar is deoxyribose C5H10O4. Deoxyribose is ribose that is missing 1 oxygen O. The 5 nucleobases are adenine A, cytosine C, guanine G, thymine T, and uracil U. Both DNA and RNA pair cytosine C with guanine G. In DNA, adenine A is paired with thymine T. In RNA, adenine A is paired with uracil U. Uracil and thymine are built on the same carbon ring with the exception that thymine has a methyl group CH3 substituted for one of the hydrogens attached to the carbon ring. There are 4 permutations to each rung on the DNA spiral: AT, TA, CG, GA. So, in a single strand of DNA, there are 4 ways to fill one placeholder, A, T, C, or G. This 4-way genetic code is a quaternary code. In binary computer algorithms, there are 2 ways to fill a placeholder: use a 0 or a 1. A computer code is a binary code. Codes or information. And what does DNA code for? DNA codes for information for building proteins from amino acids. Proteins are composed of one or more polypeptide chains. Peptides are composed of one or more amino acids. There are functional proteins that are as short as 3 amino acids long. Most proteins are more than 50 amino acids long, and some proteins are composed of as many as 30,000 amino acids. Polypeptides are peptide chains joined together by amide bonds. An amide is composed of a carbonyl group CO and an amino group NH2. It would be simpler to say that a protein is composed of amino acids. Amino acids are composed of an amino group NH3 and a carboxylate group CO2-, along with a side chain composed of carbon, hydrogen, oxygen, and nitrogen. There are as many as 500 compounds that are considered to be amino acids. There are only 20 standard amino acids that are genetically coded for by DNA. Amino acids can form in nature. Amino acids have been found in meteorites, meaning amino acids can form in space. As far as we know, the assembly of amino acids into functional proteins requires instructions from nucleic acids found only in living things. As they require a deeper context, I shall discuss the functions of lipids, carbohydrates, nucleic acids, and proteins in separate videos next.
Thank you for watching. If you enjoyed this video and want to help me make similar content, please like and subscribe.